So, by now you've heard us use the concept of statistical significance a lot. But what exactly is statistical significance, and why do we care about it? One of the fundamental principles of science is that we can never prove something with 100% certainty. Because of this, when we are designing experiments, we have to consider what's called the hypothesis and the null hypothesis. A hypothesis is an educated, testable statement that you use to create your research question. The opposite of the hypothesis is the null hypothesis. It's easiest to see the difference with an example. Let's say that we decide to perform a study asking the question, does chocolate consumption affect productivity in high school students? Your hypothesis might be that students who eat chocolate will complete a homework assignment faster than those who do not. In this case, the opposite, or the null hypothesis, would be that there is no relationship between the time to complete a homework assignment and chocolate consumption. As I said before, science cannot prove anything with 100% certainty, so statistics is used to calculate the probability of an event occurring by chance alone. This is similar to what Lauren was saying earlier with the normal distribution. It's represented by a bell curve in which all possible results are plotted against the probability of that event occurring, which is also known as a p-value. Statistics are used to test the null hypothesis, that there is no real difference, and that any differences seen are due to chance alone. Scientists have set an arbitrary probability of p less than 0.05, or 5%, as a threshold for significance, meaning that if the probability that there will be a difference this large or larger by chance alone is less than 5%, it is highly likely that the difference is real or significant in scientific terms. If the probability is above 0.05, we say that we fail to reject the null hypothesis, whereas if the probability is below 0.05, we reject the null hypothesis and it is significant. Note that we never say that we accept the null hypothesis or accept the hypothesis, because again, we can't say anything with 100% certainty. As young scientists, the statistical test that you are most likely to use will be the t-test. There are several types of t-tests, but today we're focusing on the two-sample t-test, because it is the one that you're probably going to be using most often within your own scientific experiments. A two-sample t-test is used to test if two groups have the same mean or different means. At the end of the t-test, you'll be given that p-value that we talked about previously. If the p-value is less than 0.05, it suggests that you should reject the null hypothesis, that there is no difference between the two groups. Again, this would also suggest that there is a difference, but we don't want to say that for 100% certain because it's statistics. As with most statistical tests, t-tests are affected by the sample size, or the number of samples per group. Experiments with larger sample sizes increase the strength of the test. As an example scenario in which it would be appropriate to use a two-sample t-test, let's go back to our question, does chocolate consumption affect productivity in high school students? This hypothetical experiment was conducted by giving high school students chocolate or no chocolate, and measuring the amount of time it took them to complete the same homework assignment. The mean and standard deviation for each group is listed. After performing a t-test, the p-value was 0.0111, which is below the threshold of 0.05, and suggests that we should reject the null hypothesis that there is no difference between the chocolate and no chocolate groups. Because the mean time for the chocolate group was lower, this also suggests that eating chocolate could increase productivity. Note that I did say could when I was talking here. It's important to note that it does not necessarily mean that chocolate itself increases productivity. Chocolate also contains a lot of sugar and caffeine, so both of these could be factors that are affecting the productivity. There are oftentimes alternative explanations for result, which is why it is extremely important to design proper controls in any experiment. For more information on designing controls to increase the likelihood that your results are truly a result of the dependent variable, you can watch my video on designing controls as part of our experimental design series. A second common type of statistical test is the analysis of variance, which is also known as ANOVA. An ANOVA is very similar to a two-sample t-test. The biggest difference is that while a two-sample t-test, by definition, can only compare the means of two samples, an ANOVA is used when you want to compare the mean of 3 or more different groups. As with a two-sample t-test, the output of an ANOVA is a p-value. If the p-value is less than 0.05, you can reject the null hypothesis that the means of all of the samples are the same. An important thing to know about an ANOVA is that it only tells you that there are at least 2 of the groups that are significantly different from each other, but it cannot tell you which groups are significantly different. At this point, you might be wondering why we don't just run a lot of different two-sample t-tests comparing all of the different groups individually to see if there are any that are significantly different from each other. This is a great idea, and a lot of people think that they can do that. But unfortunately, we can't actually do it, because it would lead to what are called false positives, which are events that are not in fact different from each other, but have an artificially low p-value below 0.05. If you want to know which groups are different from each other, you would have to run a Tukey HSD, which we will not be covering in this video. As with many statistical tests, ANOVA is affected by sample size. Larger sample sizes increase the strength of the test. To see how we would use an ANOVA, let's return to our original question. Does chocolate consumption affect productivity in high school students? Because we are no longer limited to 2 groups like we were with the two-sample t-test, we can now measure the time to complete a homework assignment for a lot of different groups. For example, chocolate, no candy, jelly beans, and sour gummy worms. People are going to have a big sugar high now. The p-value is 0.0001, which is less than 0.05. So we can reject the null hypothesis that there is no difference in productivity between any of the groups. It is likely that at least one group is different from the others, although we cannot identify the specific group by ANOVA. Just by looking at the means, we can see that the mean for chocolate is lower than the means for the other groups, and all the other groups are fairly similar to each other. So we can predict that if we were to run a Tukey HSD, chocolate would be found to increase productivity. We cannot say that just from the ANOVA itself. One final statistical test that you might experience is a chi-square test. The chi-square test is used when we have 2 categorical variables and want to determine if one variable is related to another. It is usually used to determine whether there is a difference between expected and observed frequencies. For this example, we're going to look at gender distribution within the US Senate. In the US, we have 100 senators, 2 from each state. We want to know whether there is a relationship between gender and being a senator. Now we will compare predicted versus observed frequencies that actually occur in the US Senate. In 2020, there are 26 women and 74 men in the Senate. This is the observed data as a raw count. But what is our expected data? For gender, we have male, female, and non-binary. If the null hypothesis that there is no relationship between gender and being a senator is true, we would expect there to be approximately equal numbers of senators from each gender, or about 33 men, 33 females, and 33 non-binary. Even before doing the analysis, it's pretty clear that these groups are not equal. The chi-square test gives us a p-value that is very small, less than 0.0001, which means that we reject the null hypothesis that there is no relationship between gender and Senate position. Chi-square tests often involve more complex data, and the expected probabilities in those cases might not be equal as they were in our example. For example, engineers use the chi-squared test to see if material has an effect on product ratings or durability. They're also used in health studies to see if there's a relationship between categorical variables in a sample population and some type of treatment. Remember, the chi-square test is used to compare observed or sample data against expected frequencies. Today, we have studied statistical analysis and why it is useful in biology. We talked about 3 different types of tests that you may be using. The t-test, which compares the means or averages between 2 samples. An ANOVA, which is similar to a t-test, but it can be used for more than 2 samples, or 3 or more. And a chi-squared test, which is different between the other 2 because it looks at categorical variables to see if there's a relationship between them, and is looking at the difference between observed and expected frequencies.
Thanks so much, and we hope you enjoy this roto video.